Thanks. So we left off at sequence alignment. So what I want to do is, the next part is to actually talk about some of the algorithms that we use to do alignments. So pretty much they can be divided into three different groups. The first is called a global sequence alignment. It's otherwise known as the Needleman-Wunsch algorithm. And then you have kind of a local sequence alignment, or the Smith-Waterman algorithm. Probably the Smith-Waterman algorithm is better known as BLAST. Essentially, BLAST uses the Smith-Waterman algorithm to do a local sequence alignment. And BLAST, in fact, is a local sequence aligner. And then you have this kind of hybrid of the two, which is called G-local sequence alignment. So we're going to go over a couple of examples. So this is an example of global alignment. What it defines is it's exactly as it sounds. It's basically full-length alignment of two entire sequences. And effectively, what it tries to do is it tries to basically just add gaps where one sequence may be shorter or longer than the other. Then you basically add gaps to compensate. So if you look here, these are two examples of two sequences where you can kind of see the global alignment here, and you can see that there's basically one gap inserted in each sequence to produce the alignment. And you end up having 12 matches over across 15 base pairs, which leads to an 80% alignment, 80% identity. On the other hand, you might have kind of a local alignment. Now, local alignment is usually better if you have sequences that are very greatly in terms of the lengths. The idea is to take a short sequence and align it against a long sequence and vice versa. So with a local alignment, essentially what you want to look for is a substring or a matched substring of two sequences. Now, unmatched bases are not penalized in a local sequence alignment. So this is an example where you basically have two sequences, and basically there's 6 matches across 7 base pairs. The other parts outside of the local region that are aligning do not actually cause a penalty, even though there are clearly mismatches there. And this method tends to be the most sensitive, more sensitive than a global alignment. This is a method used by BLAST, and this is an example. Then you have this other combination, which is called G-local. You can think of it as a combination of a global and local alignment. And the idea behind that is that the substring has to match the entirety of the query sequence. So in other words, you have to use the entire query sequence. You can't use a substring of it. You have to use the whole query sequence and match it against the substring of the reference sequence. So in this case, basically the ends of the query sequence that don't align are actually considered in the equation, and you get a penalty. So in this case, where you have exactly the same query sequence, you have 6 matches out of 12 base pairs instead of 6 matches out of 8 base pairs or 7 base pairs, and that leads to a decreased identity and decreased significance. Okay. So how do you actually choose what particular aligner to use? Well, one question is, are you aligning short sequences or long sequences? So, for instance, for many common aligners, essentially, you have short reads generated by Illumina or some other technology that then align to reference sequences in GenBank. Well, in that case, you probably want to use more of a local alignment rather than a global alignment. If you have long contigs that are generated from those short sequences that then are aligned to the reference, then you probably want to use something more global, basically. And then also you want to determine how well does it handle kind of gaps, like insertions and deletions. Typically, global aligners handle gaps better than local aligners. So what are they? Some of the short read aligners, and it's probably pretty familiar with you, but for use of like Bowtie and BWA. BWA is particularly good because it handles kind of small gaps or indels, insertions, deletions very effectively. And then there's this new algorithm called SNAP that Taylor and I have written basically, which actually appears to be faster than any of these other ones. So you have also kind of long read aligners, basically.
Can I ask a question about how those short read aligners work?
Yes.
So the algorithm, what is presumably some sort of iterative thing where they...
Yeah, it's actually, a lot of them use essentially the principle of, well, we'll actually go into that.
Okay.
But like Snap, for instance, uses the concept of a hash table. So the hash table is the best way, in computer science, it's the fastest way to actually access kind of random sequences or random blocks of data. And so essentially what they do is they kind of take a query sequence, they chop it up into like MERS or K-MERS. So usually they're like 10 nucleotides or 10 amino acids length. Then you do a sliding window across, you generate all possible 10 MERS. And then you basically insert it into a hash table. And then as you go into your And I'm sorry, that the 10 mers are actually used for, the references are chopped up. So like something like GenBank NR is chopped up into 10 mers. And then they chop up the query into 10 mers, and then you could directly compare kind of the 10 mers with each other. You could do those very, very quickly. And the reason you can do that very quickly is that these processes can be made highly parallel within certain computer architectures. So that's how these algorithms kind of go. BLAST works that way also. It uses kind of an n-mer, like a k-mer long length.
So there's probably trillions in each, or trillions in the one.
That's correct, right? And then there are different criteria as to how you call it match. You know, if it's just one 10-mer as a match, usually you get a lot of false positives because you can imagine there are a lot of like background matches and cross matches to background. So typically there are different ways you determine match. Some require, for instance, like two 10-mers in two different regions of the short read. Some people require like overlapping 10-mers and things like that. So, but there's another technique which is actually faster but less sensitive than the k-mer approach. But we'll actually go into that because certain algorithms use it. Long sequence aligners. You know, things like BLAST and BLAT can align many things can handle gaps, etc. You know, BLAT in particular handles very large gaps or introns really well. So the general idea, okay, and this sort of like gets into your ideas, is that you want to have kind of a heuristic alignment, okay, and that's kind of the k-mer theory. So in other words, you generate, you don't want to do like exhaustive alignment for large sequences. So the idea is that what you do is you generate these seeds, which are also known as the k-mers, like these 10-mer long seeds, and you want to look for perfect matches to just those seeds. So instead of aligning, say, a huge 150 base pair sequence, which would take a long time, you just align kind of short 10-mer sequences, so you kind of condense the problem a little bit. The seeding strategy, however, as you can see, has a large effect on the sensitivity, right? Because BLAT, for instance, requires two perfect nearby 11-mer matches, and it calls it a positive. So it kind of saves a lot of time, and yet it may not be as sensitive because it requires like two perfect matches, and it can't handle kind of, say a sequence that has some variation across the whole region, basically.
We ask all donors, new, old, and regular, to come to the UCSF Blood Center, Millbury Union, across the street at 500 Parnassus Avenue in room MU09, next door to the subway. Today's platelet drive is being held from now until 6 in the evening. All successful platelet donors will receive a Starbucks gift card and free parking in the Parnassus garage, and a Bear Hugs gift certificate for UCSF vendors. So please come and give the gift of life today.
Okay, so basically, there is this concept of basically doing, so let's go into assembly. I'll actually talk a little more about alignment later. But let's go into assembly, which is a similar problem. The idea, so this is for whole genome assembly. Basically, the idea is that you basically do what we call whole genome shotgun sequencing. You sort of take a genome, you cleave it into many small parts, you sequence each of the small parts, and then you assemble them together, all the fragments. And so this is the general idea, genomic DNA from, say, a human genome, shotgun, basically cleave apart to fragments, and then sequence each fragment using a shotgun approach. You can either align contiguous sequences that overlap, and then you finally end up with a finished sequence. Now, so the power of shotgun sequencing has increased with the rise of deep sequencing or next generation sequencing. And the simple idea is that you just get much more coverage. Now, the big problem, though, is that the fragment lengths are much shorter, and there's higher error rates. So how do you actually handle this problem? Well, one is that next generation sequencing, many technologies have what they call paired end or mate sequencing. And the idea behind that is that you can, if you imagine a DNA fragment of a known length or unknown length, you could sequence the two ends of the fragment of a known size. Now, that actually becomes important when it comes time to assembly. Now, the reason that's important is because you not only want to sequence and look for overlaps, you also want to know roughly where is it globally kind of fit within the genome. And this is especially important for large genomes. So, for instance, for something like 454 pyrosequencing, what you might want to do is basically generate these amplicons of extremely long lengths. So you might go for 1 kb, 8 kb, 20 kb, even 100 kb, and then sequence the ends of it. And then based on those particular sequences, you could then, for any given read, you could basically place it within the genome. And this is actually important if you want to do, say, de novo assembly where you don't actually know the genomic structure of say, a new organism. So we're actually faced with that problem right now. We're trying to sequence Naegleria fowleri, kind of the which is basically a free-living amoeba that causes encephalitis. And one big issue is that we don't know about the genomic organization or structure of the we have no reference to use. So this is an approach we have to use kind of very long, essentially long amplicons and sequence doing paired end sequences from very long amplicons to be able to do that. Now, so a lot of challenges, and some of them are the fact you have short fragments. The other issue is that you have you need a large amount of coverage, and you also have short overlaps and higher error rates. So approaches. So we'll go into a little bit of approaches, but let's first think about different types of sequencing you can do. You can do genome sequencing. You can do exome sequencing. You sequence transcriptomes. You can do clone and amplicon sequencing. Probably in terms of the near future, you're more likely to see amplicon sequencing as something which might be in the clinical lab because it's more straightforward. It's essentially sequencing PCR products, whereas genome and exome sequencing are pretty much research only for now. They're rapidly moving into the clinic, though. Especially human genome sequencing. So this is an example of kind of contig coverage. If you go across, so contig stands for contiguous sequences, and reads are the individual raw sequence reads you get from your Illumina instrument or other deep sequencing instrument. And you can imagine them as almost like raindrops on the sidewalk, where basically you're going to get uneven coverage, no matter typically, no matter kind of what genome or what template you're actually using. So the amount of coverage can be described in what they call Landor-Waterman statistics or LW statistics. Now, this is basically a means of basically determining how much coverage do you particularly need to be able to, say, reassemble any given genome. And you don't need to really know the formula. I guess just understand that you can actually describe kind of minimal coverage in using Landor-Waterman statistics. So this gives you an example using these statistics. Like, for instance, if you have a genome size of 1 megabase, your read lengths are 600 bases, and given a certain amount of detectable overlap, you can see that basically, given after a certain number of cycles, you can generate a certain number of contigs, have what they call islands. So islands are basically, in sequencing parlance, what islands are basically, they're long contigs that you can't really extend further from, and that's what they're called islands. Ideally, if you sequence the whole thing, you have only one island, which is the sequence of the genome for a contiguous genome. They may also reflect say individual genomes or genomic sequences like plasmids and stuff, where obviously you can't extend because they're not contiguous with other parts of the genome. So the two types of assembly you can do, one is a reference-based and one is de novo. A large amount of the human resequencing that's done is reference-based. George says de novo assembly has been used really for kind of novel genomes or kind of novel sequences. And these are kind of the ways that we do kind of assembly. So one's called, and this is called related because a lot of the people, the same algorithms that are used for assembly are also used for alignment. Okay, so we'll go into a couple of them because I think it's important to understand like what are the assembly algorithms out there. One's called the greedy algorithm. The other one's called OLC, or overlap layout consensus. We'll go into that. There's another method that you'll see commonly called Debrugian graphs, and that's also another variation of the Kamer kind of assembly algorithm. And then finally, there's the Burrows-Wheeler transform. Okay, so what do they mean? The first is a greedy algorithm. The idea is it's really quick. You basically take the two sequences with the largest overlap out of your whole set, you merge them, and then you repeat. You merge, repeat, and as you can see, your contigs get bigger and bigger. Essentially, it looks for the largest amount of overlaps between existing set of sequences. The problem with this is that it's highly prone to misassembly. Because I might imagine you just have like some area that has a spurious overlap, maybe because of a residual primer or something. That will basically throw off your assembly, and you'll end up kind of assembling incorrect or chimeric sequences. So it's not really commonly used. Another form is called overlap layout consensus. So the idea behind that, it's a little more, I would say, a more rational way of doing assembly than the greedy algorithm. What you do is you basically find all pairs of sequences that overlap. That's called the overlap part. Then in layout, you basically remove all of the overlaps that you would consider redundant or weak or not very, to have a lot of errors, that have low quality, and then you end up trying to merging only pairs of sequences that overlap unambiguously. In other words, what you want to do is you want to first get a general layout by overlapping kind of essentially by only using the best overlaps first, and that way you can make sure that you're generating essentially you're basically generating templates that are roughly correct or that are most likely to be correct. And then finally, you want to call the consensus base at positions where the reads overlap. So where you get good kind of overlap, you can then finally call the consensus base, or what the consensus would be. Okay. So how does that actually work? This is an example of what they call an overlap graph, where if you might imagine a vertex is basically a string, or a node is this particular string. It's like in this case we have 3 sequences, U, V, and W. An edge represents an overlap between any 2 strings. And what you do is effectively the algorithm just generates a graph out of imagine all of the reads. You generate a graph. In this case, you have 3 reads that overlap, partially overlap. So it generates essentially this kind of graph format on the computer. And the idea is that, so what you have with overlap layout consensus, the main entity is the read, and the relationship is always the overlap. So what it does is it sort of records the overlaps. And by doing so, it can then basically combine these kind of together. Once it generates the graph, it can effectively look at the graph, find the areas that have the best amount of overlap, most redundancy, do those first, and then successfully do the rest. Okay, so that's the sort of idea about it. Now, this way works well. It tends to be very, very slow, as you might imagine, because especially as your number of reads go up. This is actually the algorithm that's used by Fred and Frapp. I don't know if you know Fred and Frapp. That's actually what they do for Sanger sequencing when we didn't have deep sequencing. That was actually the method to actually assemble sequences. And this is actually the method of how the first human genome was assembled by Craig Ventnor and also both separately by the NIH. So the general idea is sort of like this. You have an overlap graph. You kind of you can actually traverse the graph and actually form contigs because it forms edges. Okay. Now here's some other approaches. So the third approach that we're talking about is the de Bruijn graph and the Euler graph. Now, this approach is an alternative to the overlap layout consensus graph. It's more suited towards short read assembly, and it's based on what we call de Bruijn graph theory. Okay. De Bruijn was basically I believe a Swiss mathematician who basically designed this. And it was implemented in Velvet. There are other algorithms. One's called Abyss. Another one's called Adena. And these another called SOAP de Novo. These all use de Bruijn graphs. Okay. So how does a de Bruijn graph differ? Well, one is that it's a particular type of overlap graph where, for instance, so as opposed to overlap layout consensus, the actual vertices can be reads or strings of any length. In de Bruijn graph, they have to be the Kamer length. So it's a similar idea to Kamers, where you're taking your reads and you're basically chopping up into Kamers. But what's interesting about de Bruijn graphs is de Bruijn graphs have an edge that's exactly of length k minus 1. So in other words, the overlap is always essentially it's a bit of a sliding window. It's always separated by kind of 1. Now, why is that important? Well, if you have like characteristics like this, it actually becomes much easier to do graph traversal and actually to generate these graphs. So it's much faster, basically. Now, why is it not as good? Well, the reason it's not as good is because it's so restricted. It tends not to be as sensitive as kind of just looking at all types of overlaps, because it has to be a very specifically defined overlap. So the idea is like here, where you have like a read of length L. Let's say you have L minus K plus 1 Kamers are generated. So you do a sliding window, and you generate all of these vertices by taking one read and sliding it across. So in this case, you have a read of length 12. You take the 9 mers, and you basically generate all possible 9 mers from that read. Okay, and then those are actually considered an edge. When you have any 2 adjacent Kamers, they're considered an edge of the graph. So you might imagine a huge graph that looks like this. And then basically by the graph method, you can actually traverse the graph, and actually you can unambiguously form contigs by traversing the graph. Where as you go from node to node of the de Bruijn graph, you're simply adding one base at a time. Does that sort of make sense, basically? Okay, so this is an example. This is kind of a de Bruijn graph where k equals 4, where you kind of see so basically, k minus 1 as you go. So as you traverse, you might imagine that you could actually traverse, like if you look at the GAC, you could then add a T to that, which then adds a G, which adds a G, which adds a G, which adds an A, and you could sort of traverse the kind of de Bruijn graph. So this gives you an example. Now, what's nice about it is it's very fast. Okay, it's very elegant. Bad about it, and the other thing is that the graph size can be huge. So for a human genome, the de Bruijn graph the human genome is about 3 gigabases in size. Once you make a de Bruijn graph out of it, it becomes 200 gigabases. However, even though
that's because you re-represent every 10.
Exactly.
Basically,
you re-represent every possible 10 in every possible sequence. Exactly. Yeah. And then that's roughly that's it. Now the so that's actually the de Bruijn graph. Now, very elegant. The disadvantage is it's not as sensitive. Okay, so for instance, some people use you can also use distribution graphs for alignments as well. The problem with that is that it tends not to be as sensitive in alignment. It's not going to pick up as much as kind of brute force methods, such as BLAST, which also uses k-mers, but doesn't represent it in terms of a distribution graph. Okay, but so finally, there's a Burrows-Wheeler transform. Now that one is also known as BWT. That actually is very similar to an overlap layout consensus. So I actually won't go into that too much. I would basically just say that it converts the reads into, it uses kind of the BW transform to convert the reads to a certain format. But otherwise, the actual algorithm itself is an overlap layout consensus algorithm. You just look for overlaps, et cetera. It just converts it to a form that's actually more efficient than just sequences. So if we think about all these three different methods, overlap layout consensus, diffusion, and BWT, well, these are kind of different purposes. For the overlap layout consensus, small genome, long reads, handles, indels, you can think of this as Sanger sequencing, because this is ideal for Sanger sequences. Diffusion graph is, on the other hand, is great for short reads, large genome, and no indels. So this is, I think of Illumina technology as perfect for it. But now it's changing a little bit as Illumina the reads are getting longer, but when Illumina used to have like 25 base pair or 36 base pair, this was ideal. And then you have BWT, the Burrows-Wheeler transform. That one's a large genome, can be either long or short reads, and currently doesn't really handle indels too well, although it's starting to do a better job. So some examples, overlap layout consensus. You don't have to memorize this, and the slides will be available. Probably Fred-Frapp is the most common. A lot of people use CAP3. These are usually used to handle a small number of sequences, usually Sanger sequences. Diffusion graph, SOAP de Novo, Abyss, and Velvet are some good examples. Burrows-Wheeler, which BW and Bowtie both use Burrows-Wheeler. So sequencing strategy then always becomes the fact is that in general you need to have about 30 times coverage. So that's a good number to remember. So for instance, if you want to sequence human genome, you need to reliably, you probably need at least 30 times coverage. Minimal, you'll need 8 times coverage. And those are because of errors and primers and other issues. But obviously need more coverage is even better. Okay. And a big issue has been this issue of quality filtering and trimming. It actually needs to be done. These kind of pre-steps, which include filtering, which include filtering by quality, trimming the ends of primers, debarcoding the data, low complexity filtering also. It makes the resulting assembly or the alignments much more easier, and it reduces the risk of doing spurious alignments or assemblies. So this gives you an example of this is kind of using a dual assembly using an algorithm called Price. Price is actually a completely different type of algorithm. It's based on starting with the seed, and then it sort of builds on the seeds. It takes advantage of kind of paired-end sequencing data or mate-paired sequencing data generated by Illumina, and you can actually generate whole viral genomes by this method.
So I have a question. So it seems like all these methods would be extremely vulnerable to areas that are like long runs of the same base.
Absolutely, yeah. Long runs. So those are what are called low complexity regions. Yeah, very hard. And the way to deal with low complexity has always been longer reads, because it's like you can handle say 5 repeats of the same sequence if you could sequence all of it, and then you know that there are 5 repeats. But otherwise. And this is why, for instance, to do essentially fixing genomes, especially genomes that have low complexity. Another issue is that genomes that have that are kind of AT or GC rich, because they also tend to have more of these kind of long stretches of single nucleotides, what we call homopolymeric sequences. Even worse, some of the technologies, the disadvantage is that they produce homopolymers that are erroneous, that can't handle homopolymers. So a good example is 454. 454 is notorious for not being able to handle more than 6 bases at a time. So in other words, if you have, let's say, a stretch of 8 A's, which is not uncommon in the human genome, what happens is the 454 will only report it as 6, 7, 8, in some cases 9 A's. So it incorrectly gives you a number of homopolymers, which is a big problem. Illumina does a much better job of dealing with homopolymers. Their errors are more kind of single base changes. But you're right, and there's no good solution to that. The best way has always been to do what they call finishing, which is basically just resequencing areas that are low complexity. So one example of kind of assembly, so this is an exact example, like, so how do you handle a true variant? And this is, one example is small deletion in a tandem repeat. Now, why is that important? Well, because some mutations, as you know, are repeats. So to know whether or not there are 3, 4, or 5 repeats becomes really, really important. And it also becomes important in the sense that you really have to know exactly how many repetitions there are. So this is an example where imagine you have a reference that has 5 repetitions of a short 7-base sequence, okay? And then your sample has only 4 repetitions, that's 1 fewer. So in other words, let's say your sample happens to be a deletion of one of the repetitions. That may be clinically significant. So how are you going to be able to actually deal with this or detect this particular variant? Well, one big issue, and this is kind of the error that the problem that you pointed out, the alignment of just short reads alone may not show that deletion. Okay. Alignment reads to the reference, they perfectly covers the reference with no more than 2 errors. So in other words, you have a reference that has like 5 copies of 5 repeats. Your actual, basically your short reads, they're not enough. Your short reads are generated for something with only 4 repeats. They're still not enough because you could align it fairly well across the 5 repeat reference. So in other words, you would miss the fact that your actual sample only has 4 repeats. Okay. So how do you actually do that? Well, one is if you do proper assembly, you can actually assemble the reads, assuming there's enough overlap. You can assemble reads enough so that they finally do show the 7-base deletion, and that is to some extent dependent on the length of your reads and quality of your data. Okay. So how do you visualize this? I'll kind of skip pretty rapidly through this. Basically, the most important thing to remember is that the most common format now is called the SAM format, which is the sequence alignment, basically.
SAM tools?
Yeah, SAM tools use the SAM format. BAM is a binary version of SAM, so BAM is the same as SAM. I think you'll probably end up seeing SAM being used more. It's becoming more and more standardized for looking at data. And then you have, I don't know if you're familiar with this. This is called the Integrative Genomics Viewer. It's a way to actually look at the reads and as they're mapped on. There are other tools that have been developed commercially, things like Genius, for instance, which allow kind of biologists to be able to handle and manipulate sequence data. And then this other term, have you heard of N50? Right. Okay, so this is actually important because it's a good measure of basically the quality of your assembly, okay? And the idea is that you want your N50 as large as possible, okay? What it's called, it's actually a weighted median of the lengths of the contigs. So for example, let's just say you have assembled genome in 5 contigs. There are 100, 300, 500, 700, 900 kilobases in length. The genome itself is the sum total of that, 2,500 kilobases. So it turns out that half of your genome, or more than 50% of your genome, is assembled in 2 contigs, right? 700 and 900. That's because if you basically sum up the contigs from largest to smallest, when you add 700, 900, you get 1,600. That's actually more than 50% of the size of the genome. So that's the N50 is the length of that smaller contig. So in this case, the N50 is 700 kilobases. Okay. I actually have an example in one of these one of the quizzes. So why is that important? Because as the N50 gets larger, basically, that is a measure of quality of assembly. And in fact, if you look at any of these papers that do assembly of genomes, you'll see they quote the N50 basically for all of their data. Okay. I'll finally go into like some of the challenges of what they call transcriptome profiling or transcriptome sequencing, and it's also commonly called RNA-seq by Illumina. The idea behind it is that you essentially do reverse transcription of your RNA, you fragment the cDNA, you do size selection and sequencing of each end. Now this is a little more perilous than genomic sequencing, and the reason is because as you know with RNA you have splice variants, right? So the problem with that is that you can't. There's no good reference, right? Because unless you have a good transcriptome library, but by just mapping reads against a genome, you actually may end up with like false or erroneous mappings because it doesn't take into account kind of introns that exist as well as kind of splice variants. So there's no correct way to analyze. So there are 2 major branches. One idea is to directly align the reads to a genome, splice and unsplice to a gene. And then the other method is basically take your transcripts, assemble them first, and then align the transcripts to the genome. Okay, so let's go into that. So let's talk about some problems about that. Well, one is that if you just align the reads to exons, one issue is that you actually don't handle junctions very well. Okay, the splice junctions. So the idea is that what you need is that you need one good way to correct that might be to have better alignments. This is an example basically where you have the alignment artifacts of the splice junction introduces false positive SNP calls. But if it's within the, if there's a good alignment kind of within the exon, then you can find kind of truncating mutations in tumor suppressor genes. So this is where it becomes important to have kind of proper alignments. So how do you actually handle this issue of exon junctions? Well, one is to basically take your reference genome, like a human genome, append all of the known exon junction sequences to it. So that's a quick way. Or you can just simply generate a human transcriptome, basically. And now that's fine with something like the human, where you sort of know roughly what the transcriptome looks like. But it's a big problem for, say, other organisms where you don't even know the transcriptome. If the genome hasn't been sequenced, some organisms the genome hasn't even been sequenced. Even in organisms where the genome's been sequenced, you don't know what transcriptome looks like. So good examples like Staph aureus. We sequenced like 50 Staph aureus genomes. But no one's actually sequenced the transcriptome of Staph aureus, so no one knows exactly, exactly what's going on with that. So one issue is basically to align the genome and transcriptome separately, and then kind of compare the alignments. Another possibility is that there's certain aligners that will automatically do what they call splicing. It'll kind of calculate what are all possible exon junctions, because if you recall, splicing, like RNA splicing, takes into account specific rules with regards to nucleotides. So calculate all possible, exon junctions and do it for you. So the program called Top Hat, which is commonly used, is called Top Hat and Cufflinks, which is probably commonly used for this. It's called a spliced aligner. Another possibility is also to do your assembly first before you do any mapping or alignment. These are kind of some of the errors that pesky errors that you might see. One is like a recurrent RNA editing. You can see junction reads that are misaligned or skewed expression. These are common kind of challenges that you approach when you're trying to sequence tumor RNA versus sequencing tumor DNA. There are ways of also generating expression data from alignment. There are a lot of these packages that you can use for that. But ultimately, this is a very powerful strategy. Like this is an example. This is a paper published in Nature where they found that they were able to use transcriptome profiling and actually found a common translocation that was found in lymphoma, and they were able to find it because it turns out that the RNA product was actually a spliced gene product from essentially two distant regions. So it's kind of a neat way to actually, by interrogating the transcriptome and interrogating the genome, you can find kind of these translocations and other changes that reflect such a cancer. Okay.
The transcriptome and exome are the same thing.
They're the same. Yeah. So some programs. Okay, so that's basically it for that. Do you have any questions about that, basically? Okay.
Nope.
Just that part of it?
What? Let me just see what it is.
Yeah, I want to get it. That way I can move into this.
Oh, it's just Jillian. She, I guess there's a smear on a new leukemic. That's why she's not here.
That's leukemic. Okay. Okay. Okay, so let's go into some specific applications now. So I'm going to talk to you here about, the focus is really going to be on kind of microbiological applications, although you can see there are a lot of like cancer applications as well. But those are kind of, I would say, the two major areas now that there's some interest in using kind of genomics in lab medicine. But one is, you saw this yesterday, bioinformatics challenges. We know it's growing very rapidly. We need to have a scalable computational backbone. Part of our solution has been to sort of adopt like cloud computing methods. So we're taking advantage of basically technologies that have already been kind of harnessed or honed by the tech industry. We have a grant. Actually, I have a grant from Amazon.com to actually do this, put this on a cloud computing type framework. So one thing that we've been using is a hypertable database. So instead of storing the sequences flat files, we actually store them in kind of a Google-like database. It's basically based on Google's Bigtable, which is exactly how. So the idea behind that is called distributed computing, where imagine. So basically, if you think about it, how does Google actually enable you to just put anything into a search engine and basically within seconds give you a result? Well, they don't actually store. So they actually store they have somewhere in the order of now 100 petabytes of data. And they don't store all of their data in, say, one central location. They actually store it in distributed. Yeah. They actually don't store their information in, say, one location. They actually end up storing it across the world, basically. But most of the places are in the United States in large server farms. And the idea is that basically, in terms of accessing the data, the data itself is sort of not recorded in terms of like essentially files. They're actually reported in kind of this distributed database that will dynamically kind of allocate more space as it's needed. So imagine Google basically not only saves like current data, but kind of saves references to old pages and constantly kind of add on and on. It's a similar idea to it's basically the idea of cloud-based storage in that as a server gets filled, then the algorithm will automatically kind of go on to like open and kind of equalize the data within different areas. Okay, so this is how this is sort of how Hypertable works. So for instance, like right now, our Hypertable database, it's not stored on a single computer. I actually have it stored across 10 different machines. They're all at Mission Bay, basically. So 10 different machines on, say, the server farm. This is at the QB3 computational cluster at Mission Bay, where you can distribute the data across any number of machines. But let's say that it starts getting filled up, and let's say I need to put in more than 10 machines. Well, in theory, you can dynamically, I could just add, say, 5 more machines from China Basin, 5 more machines from Parnassus. You could do something like that in order to store and keep your data, basically. So we have now over like 10 billion sequences now, and this is just from MyPath, basically, generated from that, and that's all being stored in Hypertable now. So now with customizable analysis modules, so basically there are various programs. The problem right now is that there isn't like one kind of essentially user-friendly bit of software you can use to do a data analysis. So what people have done together, they cobbled together all of these open source tools that have been developed. There has been some attempt to sort of bundle these tools together. So one idea is to like basically commercialize kind of a GUI, a graphical user interface, like commercialized software where you could just run the tool. So a good example of that is Genius. Another example is CLC Bio, Galaxy. These are basically software packages from biologists. And essentially what they do is they simply run these tools, which are provided for free, which are open source tools. So you don't have to be a bioinformaticist or programmer to be able to run these tools. But you kind of see the same players. For like assembly, you see Price, Adina, Abyss, Velvet, Soap de Novo. For map align, there's Blast and Blatt, Soap Aligner, Mac, transcript analysis, Top Hat and Cufflinks, Panther, buried detection, you can use SSAHA, Saha, or Mac, and then multiple alignment, you can use Muscle or ClusLW. So for different purposes, you can kind of do different analyses. And they can generate these pipelines. So one pipeline might be you might want to align things against MRSA to resequence MRSA. Another pipeline might be to look for new viruses and hepatitis. So different applications. We actually have one for pathogen detection discovery. It's listed here. We kind of went over that yesterday. One big issue has been kind of the time it takes to actually do this. So the nice thing about going to a cloud and clusters is that you can use multiple cores. So the idea is that you can, for 100 million sequences, it would take about a month to analyze on a single top-of-the-line machine. You get down to somewhere in the order of 6 to 12 hours by putting it on the cloud. And Taylor and I have been working on some new algorithms. We have an algorithm called SNAP now, called the Single Nucleotide Alignment Project, done in collaboration with the computational group at UC Berkeley. And we actually, it has a capacity now of aligning 100 million reads per minute. So we actually timed it yesterday. It's 100 million reads for 47 seconds. Wow. Across the synthetic data set. It's really kind of neat, basically. So the idea is that we could actually assemble the human genome from, say, raw data. I think within like 30 minutes, we can assemble a whole human genome, basically. So we're trying to see, and this is the nice part is also you don't have to use a large. I mean, it is a large machine. It's a 48-core machine, but it's a single machine. So it's something that's clearly accessible by say clinical laboratories. Would be the idea. We also have this other tool. This is a tool developed by the Los Alamos National Lab, actually, that we're collaborating with. It's called Sequidex, and Sequidex is meant to be a detection tool. So what it does, it uses the same kind of k-mer theory, but imagine instead of using it to align, it generates k-mers. It generates k-mers of databases. So the idea is, let's say you want to identify a bacteria or a virus. What you can do is you could list the database of all viral proteins in RefSeq. You could then generate peptide k-mers and then run it through this program and then rapidly scan, say, raw deep sequencing data to identify what signatures might be present. So the idea, what's nice about this is that this tool can actually go through 1 billion reads a minute, basically, on a server. It actually goes through the same billion reads in like 20, 30 minutes on a laptop. So we actually have this running on a laptop now, basically. And the idea is that this gives an example. We just gave it like metagenomic data from diarrheal disease. So these are basically pooled samples from 10 children with diarrhea. We did raw deep sequencing of them. We generated 100 million reads. You can see here it detected basically these like 4 viruses very quickly, basically. And then these are all confirmed to be correct, basically. So adeno-associated virus, RSV, sapovirus, which is a Kalichi virus, and human enterovirus. So the idea is, like in the future, could you like run this on your laptop and basically as a way to do quick interpretation, say for clinical labs for identifying for pathogen identification, would be kind of the application of this.
Although with the mass spec approaches, those are already pretty fast for pathogen identification.
They are, but they tend not to be very specific, and they tend not to be broadly based as these, right? And they also have a problem with kind of detection. So, but they do work pretty well. So, like there's an IBIS instrument which is able to identify like a panels of like 8 different organisms. One other issue though with that is that they're not really typically amenable to like small clinical labs.
Sure.
Because they're not like portable. Whereas with this, you can have like portable benchtop sequencers, which is kind of the promise of this type of thing. yeah, it probably isn't. Okay, so let's go into like just a few more applications. One is called the 2009 H1N1 influenza virus. I can tell you that we were able to do this. We were able to identify, for instance, the existence of strep pneumo co-infection, which is considered prognostically important for H1N1. We will also show that the more you sequence, the greater their sensitivity for detecting virus. So it actually turns out that the percent of deep sequencing reads that you actually get to a virus is linearly proportional to the viral titer. So basically, as you sequence more, you can detect things at lower and lower viral titers. In fact, we were able to detect things as low as like 17 copies of the virus, basically so it turned out to be 18 reads out of 110,000 or 270 reads out of 2 million. Basically. We're also able to show that with this type of study is that taking a raw nasal swab, you can actually assemble like the influenza genome. So this is an example where we basically assembled about 90% of the genome de novo. We didn't use directed assembly. We basically removed all flu sequences from the database and then assembled it without using any reference. And you can sort of recover kind of a novel genome that way. Let me give you another application. This is molecular typing. One big issue has been like, can we type, say, EHVISA or MRSA? HVISA stands for heterogeneous vancomycin intermediate Staphylococcus aureus. You see here that clones that reduce susceptibility to vancomycin. So what we're doing is we're actually doing whole genome sequencing of these HVISA strains. Traditionally, it's been done by SPA typing. Do you know what SPA stands for? The Staphylococcus protein A. So basically, it tends to be highly variable among different strains, so it tends to be pretty strain specific. But it's probably better to actually look at kind of a whole genome approach, because then you can identify, for instance, resistance determinants or mechanisms of resistance. In fact, we're actually working with Chip Chambers. He's actually identified an MRSA strain that's resistant to ceftobiprole, kind of the latest generation of 5th generation cephalosporin, ceftobiprole and ceftaroline. He's actually identified a strain that's resistant to that. And we've actually identified it's another change. But we did whole genome sequences isolates, and we identified to another kind of interesting change in the PVPs, as you might expect. It has some binding proteins, basically, of this particular strain. So it's actually a pretty scary strain. He has one that's resistant to vancomycin, daptomycin, and ceftobiprole now. So I'm trying to look at kind of mechanisms of resistance. No, he generated in the lab, actually. So he generated, he basically passaged it,
Maybe you shouldn't do that.
I mean, you don't want that thing getting out there, basically. And that's, But so another application is transcriptome profiling. So this is a study that we're doing with Johns Hopkins, where we're trying to look at basically patients that have chronic Lyme disease and attempting to identify what is the gene expression or host response in the setting of patients that develop post-Lyme syndrome. So this is a kind of the controversy. The controversy is that, could it be caused by other pathogens that they're known to cause Lyme? Borrelia burgdorferi causes the cause of Lyme. Could this be maybe a co-infection with anaplasma, with babesia? Could this be a host immune response that may be causing post-Lyme syndrome? So our plan is actually to do transcriptome profiling. We're using kind of the top hat cufflinks strategy. And I can tell you that some of the findings, like for one is that we actually found that, so these are kind of, this is the summary of about 30 years of work done on Lyme disease host response. You see these are kind of all of the cytokines and other genes that are upregulated or downregulated in the setting of acute Lyme infection. And you say what we did is we did basically, we only did 2 patients. We put this on a MySeq. We got the data really quickly. Then we did a transcriptome analysis globally. And to make a long story short, I can basically tell you that we were able to basically reproduce a lot of these. In fact, we looked more deeply. We actually reproduced almost all of these. So all of the known kind of factors involved, cytokines involved in Lyme disease response, we were able to reproduce with a single run from a patient, basically. These actually were done. These were identified actually by in vitro. So there actually has not been a true transcriptome analysis done of acute Lyme in the setting of in vivo in the setting of patients with acute Lyme. So this is kind of, but what's kind of neat though is that in addition to these, we actually found 5 additional proteins that were not thought to be involved, and I actually don't know where they fit in the pathway, but presumably they must play some role in the response. So this is kind of a global way of looking at host gene expression, basically. There's also obviously an application of pathogen discovery. So, and this is motivated by the fact that we've seen lots of emerging pathogens over the past 10 years. I don't know, have I gone over this story before?
Is this with chikungunya virus?
This is with the new rhabdovirus. But somebody say we identified a new rhabdovirus. I want to say from the point of view of bioinformatics, though, this is kind of the power of it. One is that we were able to assemble. So this is where we use price. So this rhabdovirus is so divergent, it has no similarity to any other known virus. So the way to identify it is we identified one read that had remote homology to rhabdoviruses, and then we were able to assemble the genome without using a reference from one serum sample. So this is an idea where we're able to assemble it without using any reference. What makes it also interesting is the fact that subsequently we used Sequedex, whereas all of the prior work to identify and characterize the virus took about a year. We now ran it through Sequedex, and if you run the raw data, you pipe in 280 million sequences through Sequedex, you can immediately detect there's one peak, and that peak actually corresponds to rhabdoviruses. So it's kind of neat that you could actually detect a novel virus, presumably within minutes. And so what we're trying to do is we're trying to actually market this to the DoD. I'm trying to get the DoD to fund this tool so that you can use it as kind of rapid pandemic response, right? Because if you have a new pandemic, SARS coronavirus took about 4 weeks to culture and identify. So if you may imagine that if you have a new pandemic, you really don't have that amount of time. So you need to identify it really within hours to days. And I'm trying to kind of market this as a way to potentially identify new pathogens, within that time frame, basically. Okay, this is another virus, the TD monkey adenovirus. I think we'll kind of skip that. I want to end by actually talking about some of the challenges, though, because you might imagine that deep sequencing is great. These are new bioinformatic tools are fantastic, but there will be significant issues if you're thinking of introducing them into the clinical lab. And one of the big issues is that typically these are very broad techniques, and so one thing for clinical validation, it becomes problematic because especially for things like microbial identification, is that you can never really get enough good test examples to do the validation. So this was a big problem with, say, for Luminex when they tried to market their instrument for detecting respiratory viruses. They couldn't get enough coronaviruses. They only got like 10 coronaviruses, and the FDA required them to get 30 examples of natural coronavirus infections. And they weren't able to get that, and as a result, they never received FDA approval for detection of coronaviruses. Although they did get CE mark approval because in Europe, I guess the requirements were less, basically. But this is going to be a big issue, and the FDA is now considering perhaps not requiring actual prospective samples collected in the clinic to be used for validation, but maybe considering archival tissues or archival samples as a way to kind of as a surrogate for doing validation. Another thing to keep in mind is with respect to all of these is that these diagnostic devices, to get FDA approval, you must not only approve the device, you actually now have to separately get approval for the software algorithms. So in other words, you need to actually separately validate all software algorithms, especially for complex MIA or multivariate index assays. Okay, so we're talking about all the software used for bioinformatics analysis. That by itself will also have to be validated if we are thinking of actually bringing this as a clinical lab, not only the test itself and the test characteristics. Okay. And then finally, just like any other assay, you want to have an assay that has actionable results, right? You don't want to design an assay for the purpose just because it happens to be sexy or broad. You want to make sure that it actually has clinical impact. And then, we consider clinical utility. We're working on a couple things at UCSF. One's panels for virus detection, especially in immunocompromised patients. Steve Miller and I are very interested in looking for bacterial fungal species ID from culture, because that's a big issue in the microlab, is that you have something that grows, especially something that grows very slowly or it's fastidious. And right now, even identifying biochemically, you can either identify it biochemically, which takes time, or you can send it out, which comes with a delay. Something which is kind of a natural application of sequencing would be to rapidly identify known positive cultured organisms. Another thing, interest has been in HIV and hepatitis C quasi-species or whole genome sequencing, which has been of interest, especially for identifying sort of resistant mutations and patterns of evolution.
It seems like for viruses, would be a really potent technique, also because there aren't as many protein components. Whereas for bacteria and fungal, it seems like, I went to Eclipse this year, and there was a whole breakout session on the multi-TOF approach, and it seems really powerful and also very expensive once you have the machine. The machine isn't even that expensive; it's like $200,000. But then after that, with tech time and everything, you're looking at like less than $3 a sample. So it's really nice, and it's less than 8 hours to get your results. So at that would point,
it's very powerful, basically using mass spec.
Compare it to sequencing, which is like $30 to $50 per patient result.
Right, and the question is it going to get lower? Basically, it's going to be the issue.
Absolutely.
But and it's unfortunate though that the problem is like I think it's upfront costs that's made it difficult to kind of adopt. Like, because I know we would definitely be able to use, say, a mass spec instrument. The problem is like there's no money to pay for it. Rapidly, if you want to get money from the hospital, you have to justify. There has to be a return on the investment, an ROI, basically.
Right.
But it's kind of hard to do unless you can show that it's going to make money. Basically,
I just figure that with that, if you compare, that's cheaper than even doing just the long biochemicals. And so if you could replace long biochemicals, the majority of your sequencing, other than for very uncommon organisms.
Correct.
Now you're saving per patient
a lot,
huge amounts of money.
Yeah, if you think about it, these techniques they definitely have potential. Given the amount of money that we spend just on doing workup of a pneumonia in ICU patients, it turns out to be somewhere in the order of like $10,000 to $15,000 just for the microbial workup, basically alone. If you imagine, a lot of it might be replaced by a far cheaper or broader test would be kind of be the application of this. Yeah, great. So let's quickly go over some of the stuff in the quiz. So I actually have an applications quiz. Can you get a copy to Jillian as well? And people that
try.
If you have a chance.
I will do my best.
Okay. So
things get lost.
Things get lost, basically. Things get lost.
Disappear in my bag.
Okay, so let's see. So what's the difference between global and local versus glocal?
So it seems mostly like they handle gaps and read length differently.
Yep. So name at least 2 short read aligners.
Short read aligners. So BLAST is one of them.
Mm-hmm.
And I don't remember another one.
So something like SOAP, for instance, long read aligners. What's a heuristic alignment algorithm?
I don't remember.
Based on seeds, seed-based. So seed-based. Pyridine sequencing.
So that's where you have one good quality sequence and you read it from each end, and that's where you start.
Statistics described for coverage.
There's like an N50.
Lander. This is a Landerman. This is a Landerman. How about the greedy assembly? So I think you know that. OLL, De Bruijn, Borel-Willer. What's the minimum coverage required to assemble genome de novo?
30x.
Perfect. You've assembled the genome in 7 contigs: 100, 200, 400, 700,000, 2,000. Total genome size is 40. What's the N50?
So the N50, it's 1,000, right?
Perfect. What sequence line is most standardized at this point?
Sequence line format. SAM?
Yes. 2 of the 4 solutions are dealing with transcriptome mapping exon junctions.
You can add all the different exon ends, or you can trim them off.
Perfect. What is the cause of false positive calls in transcriptome? Well, we went over that. Yeah, those are errors. So rank the sequencing technology, the read throughput. Cost per read.
So for read throughput, the massively parallel is the best, followed by capillary, followed by gel.
Yeah, the other way around for cost per read. The rise in DNA sequencing capacity is outstripping what famous law by CEO of Intel?
Moore's law.
Yeah, name an open source implementation of Google's Big Table.
That's
the hyper table that we're using. Yeah. What is Amazon.com's cloud computing? I didn't actually go into that. It's Amazon's cloud computing is EC2, Microsoft's Azure. With respect to raw sequencing data, we went over that. Removal of primers and complexity filtering and quality filtering. Let me go. So we can go over that. A lot of this is, I think we've actually been over. How about this? 27. According to the FDA draft guidance, what must be separately validated along with the comp device?
Software.
Yes. Name at least 2 sequencing-based diagnostic assays that would be suitable for a CLIA lab.
That would be suitable for the CLIA lab. Novel pathogen detection. And, like a respiratory panel.
Mm-hmm. Yeah. Identification of known pathogens, a panel of pathogens. Great.
So I was just thinking, so there's this paper came out pretty recently that was looking at some of these myeloproliferative neoplasms, especially MDS, specifically refractory anemia with ringed sideroblasts, where they showed that looked like one of the main defects is an accumulation of essentially erroneous RNA transcripts. So is that something that you could look at with like an exome sequencing approach?
Certainly. Yeah, certainly. They actually have there's a lot of interest in like sequencing like various RNAs. Like there's a new thing that just opened up like siRNA sequencing for short reads, mRNA, exome sequencing. That would definitely be amenable to it. Yeah. Yeah, especially because they're short, right? If they're short reads, that's a perfect application of that. Basically. Cool. So, yeah. So if you're interested, let me know.
Yeah,
the other thing is that
these areas that I think is extremely interesting, but I like math and computer knowledge is like frighteningly small. I know so little about it and understand it as such a fun, like it's such a, it's like a third grade.